Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF entrance exam question paper conducted in the year 2017. I have divided into two parts and this is the part 1. Coming to the first question. For a single substrate reaction, doubling the substrate concentration increases the rate by tenfold. The order of the reaction is. So we know that rate of a reaction is equal to Ka raised to N, where K is the rate constant, A is concentration of substrate, N is order of the reaction. So substitute and you will get value between 3 and 4 because R is equal to 10, 2 raised to N. So N is equal to 2 raised to 3, that will be equal to 8, or 2 raised to 4, it will be equal to 16. So that value is between 10, 8 and 16. The value which lies between them is 10. So the value will be between 3 and 4. So our answer is option D. Then next one. The enzyme reaction follows Michaelis-Menten kinetics. What will be the reaction velocity at a substrate concentration Km by 3? So substrate concentration they have given as Km by 3. Substitute in the equation. V is equal to Vmax concentration of S by concentration of S plus Km. So instead of for concentration of S, you have to substitute as Km by 3. Then simplify and you will get the answer Vmax by 4. Answer is option D. What will be required volumes of 1 normal HCl and 4 normal NaOH to prepare 1 liter solution of pH 7? So they have given normality. So normality of HCl is 1 normal, that of NaOH is 4 normal, and total volume is 1 liter. So N1 V1 is equal to N2 V2. So V2 is equal to 1 minus V. Then you solve for V, you will get as 0.8 liters. Convert it into ml, that will be 800 ml. And then you have to go for the volume of NaOH by using the formula V2 is equal to 1 minus V, that will be 200 ml. So answer will be 800 ml HCl and 200 ml NaOH. Answer is option B. Then, in a bacterial growth experiment, the concentration of cells increased from 10,000 cells per ml to 30,000 cells per ml in 3 hours during the exponential growth phase. The doubling time of bacteria, so generation time or G is equal to time by number of generation. But we are not given number of generation. So you have to use the formula G is equal to T by 3.3 log N by N0. Substitute and you will get 1.9. Answer is option D. Then a 20 mer DNA contains A, G and C only. In how many ways can this DNA sequence be constructed? So it can be constructed in 3 raised to 20 ways. Answer is option C. Then match the matrices in group 1 with appropriate gradient elution conditions from group 2. So DE, AE, cephacil, that is increasing concentration of sodium chloride. Phenylcephalose is decreasing concentration of ammonium sulfate. Chromatofocusing is pH gradient, and Cephadex G100 is isocratic gradient. So the answer is 1Q, 2R, 3S and 4P. Answer is option A. Then the activity of enzyme X, total volume 5.3 ml is 2.34 micromoles of product formed per minute. The total protein content of this solution is 0.8. So what is the specific activity? So to find out the specific activity, you have the formula enzyme units per ml by protein concentration in mg per ml. So enzyme units per ml is 2.34 divided by total protein content is 0.8. So that is 0.8. So 2.34 divided by 0.8 and you will get 2.925. That will be approximately equal to 2.93. Answer is option A. Then next one. Match the techniques in group 1 with the applications given in group 2. Salting out. Salting out is protein precipitation, ultracentrifugation, sedimentation coefficient, dialysis, removal of low molecular weight impurities, isoelectric focusing, PI determination. So the answer is 1Q, 2R, 3S and 4P. Then what would be the number of protein molecules present in 1 milligram of protein having a molecular weight of 25 kDa? So molecular weight is 25 kDa or 25 kilodalton, that is equal to 25,000 dalton. And 1 dalton is 1 gram per mole. So 25,000 dalton is 25,000 gram per mole. So 1 mole of protein weighs 25,000 grams. 1 mole of substance has 6.023 into 10 raised to 23 number of molecules. So number of molecules in 1 mg or 10 raised to minus 3 grams is 6.023 into 10 raised to 23 into 10 raised to minus 3 divided by 25,000. So that will be equal to 2.4 into 10 raised to 16. I made a mistake over here. This is 16, not 6. Okay, so answer is option B, that is 2.4 into 10 raised to 16. Then purification data for an enzyme is given below. What is the fold purification? So specific activity of the first one, that is cell-free extract. That will be 150. That will total enzyme activity divided by total protein in milligrams per ml. So 150 divided by 100, that is 1.5. Specific activity of nickel NTA chromatography, that is purified sample. So 120 divided by 10, that is 12. And purification fold is specific activity of the second one divided by specific activity of first one. So 12 divided by 1.5, that is 8. Answer is option A, 8. Then when DNA is extracted from bacterial cells and analyzed for base composition, it is found that 38% of the bases are cytosine. What percentage of the base is adenine? So 38% of cytosine means guanine is also 38%. So both combined together you will get it as 76. Now A plus T is 100 minus 76, that is 24. Now A is equal to 12 and T is equal to 12. So answer will be 12%. Answer is option A. Then equal volume of cell suspensions of Escherichia coli, Saccharomyces cerevisiae, Staphylococcus lactis and Mycoplasma pneumoniae all have the same OD of 600 nanometers, that is 0.5. Which cell suspension would have the minimum and maximum number of cells respectively? So if a cell is smaller in size, then the number of those cells will be more, and if a cell is larger in size, then the number of those cells will be less. That is, size is inversely proportional to the number. So mycoplasma is having the smallest size, so maximum number of cells will be for mycoplasma, and Saccharomyces cerevisiae is having largest size, so minimum number of cells will be for Saccharomyces cerevisiae. So the answer is Saccharomyces cerevisiae and Mycoplasma pneumoniae. Answer is option A. Then next one. The spectroscopic method for detection of functional group is. So it is FTIR spectroscopy. Answer is option B. Match the native microbial source in group 1 with products in group 2. Leuconostoc mesenteroids. So that is dextran, Lactococcus lactis, Nisin, Brevibacterium brevis, Lysine, Penicillium roquefort, that is cheese. So the answer is option C, that is M3N4O1P2. Then match the enzyme in group 1 with the application in group 2. So pectinase is juice clarification, papain is meat tenderization, Glucose isomerase is high fructose corn syrup. Beta galactosidase is lactose-free milk products. So the answer is M3N4O2P1. Answer is option B. Then histone code implies which of the following? So the histone code, it is a hypothesis which states that DNA transcription is largely regulated by post-translational modification of histone proteins. Through this mechanism, a person's phenotype can change without changing their underlying genetic makeup. So it is meant for controlling of gene expression. So histone code implies which of the following? It involves modification of histones. That is why there is no change in the underlying genetic makeup. So you modify, there is post-translational modification of histone proteins. So the answer is option B. Then a plasmid of the A260 of a plasmid solution after 100-fold dilution is 0.2. Given that A260 is 1, represent 50 microgram per ml of DNA and the total volume of isolated plasmid solution is 50 microliter. What will be the concentration and the amount respectively of the isolated plasmid? So 1 OD represents 50 microgram per ml of DNA. So 0.2 OD represent 0.2 into 50 divided by 1. That is 10 microgram per ml of DNA. DNA present in 50 microliter of solution. That will be 10 into 50 divided by 1000 for micrograms per microliter. That will be 0.5 micrograms per microliter. The solution is diluted 100-fold. So multiply 0.5 by 5 into 100, that will be 50 microgram per microliter. So that is the actual amount of DNA. Now concentration will be 10 by 1000 into 100 micrograms per microliter, that is 1 microgram per microliter. So the answer here is what will be the concentration and amount respectively. So 1 and 50. 1 microgram per microliter and 50 micrograms per microliter. So answer is option. Then next one. A gene was cloned into a unique Hnd3 restriction site present in the ampicillin resistance gene of a vector that contains both ampicillin and kanamycin resistance gene. To select for only recombinant clones, the transformation mixture should be plated on which of the following plates? Now the ampicillin resistant gene became non-functional upon cutting it with Hnd3 and inserting the gene of interest. So option A, ampicillin containing plates. The recombinant will die. So that is not the answer. Then ampicillin plus kanamycin containing plate. There also the recombinant dies. Now ampicillin containing plate followed by replica plating on kanamycin containing plate. Ampicillin containing plate, the recombinant will die, so it is not possible to recover the recombinant from the ampicillin plate. So even if you do replica plating on kanamycin medium, it will not grow because there is no recombinant which is alive. So the next will be kanamycin containing plates followed by replica plating on ampicillin containing plate. Recombinants will survive on kanamycin plates. These recombinants are further checked for the presence of the gene of interest by plating on ampicillin plate. Recombinants will die on ampicillin plate. Then you compare it with the master plate, that is, and you can check which of these were the ampicillin sensitive colonies, that will be your recombinants. So answer is option D. Then from among the options given below, RNA polymerase 2 transcribes which one of the following? So it transcribes microRNA. Answer is option C. Then a student sequenced a DNA using Sanger's method and obtained the following autoradiogram. The sequence of DNAs. The smallest band will be the 5' end. So G will be the 5' end. Then you have got A, then again an A, then T and C. So GAATC. Answer will be option B, that is 5 prime. GAATC 3 prime. Then next one. The melting curve of a DNA solution is represented below. It suggests that. So we can see from the graph that at the beginning point of temperature, 1 minute. At the beginning point of temperature, there is some value for absorbance as the temperature increases. So first it is constant, then there is some value for absorbance as the temperature increases. That is this line. The absorbance remains same, and at some point of temperature the absorbance increases. So again it becomes constant, then again increases, and then again it becomes constant. So DNA is double stranded, and as temperature increases the bonds break and nucleotides are getting exposed, and because of that absorbance increased. Absorbance increase until certain point and then it become constant. Again after a certain temperature the absorbance increases and then comes to a constant value. That is what I have explained now. That is, as the temperature increases the absorbance is constant, then it increases up to a certain value of temperature, then again constant, then again increase, and then again a constant. So there are certain reasons why DNA show melting at some temperature point. So we can say that there are 80 base pairs which are double bonded, and as temperature increased they couldn't maintain hydrogen bond between A and T and they break off. So the absorbance increases till all the 80 bonds are broken and then it becomes constant. So this region, it is the region where the 80 bonds are present in the DNA, and these break, and they break until all the 80 bonds are broken, and after a particular temperature they become constant. Now as the temperature increases there may be GC contents that start melting at higher temperatures. So as the temperature increases there may be GC contents that start melting as the temperature increases. That is why this curve, no, this slant or slope. So this graph suggests that the DNA has two structures with a biased base composition. Answer is option B. Now option D, if you say that DNA has got an unbiased base composition, then you would have got a straight line. Now if it was option C, the given DNA has three structures with a biased base composition, but here only two are there, 1 and 2. There is no third one is not here. A slant is not over here. So it has only two structures. So answer is option D. B. Then next one. The DNA binding domain of a transcription factor that is specifically required for the regulation of gene A is exchanged with the DNA binding domain of another transcription factor that is required specifically for the regulation of gene B. This chimeric transcription factor will regulate. So what they have told, the DNA binding domain of a transcription factor that is specifically required for the regulation of gene A is exchanged with the DNA binding domain of another transcription factor that is required specifically for the regulation of gene B. So if gene A is exchanged with gene B, the DNA binding domain. Okay, now this chimeric transcription of the transcription DNA binding domain of transcription factor. Okay, this chimeric transcription factor will regulate gene B only. Answer is option B. Then next one. Protein synthesis in eukaryotic cells terminate at stop codons because there is no naturally occurring tRNA with an anticodon corresponding to the stop codon. So answer is option C. A DNA fragment was digested with a restriction enzyme X with the recognition sequence. that would cleave at positions of the arrow as shown. You want to clone this fragment in a vector which does not contain the restriction site for X. Which one of the following enzymes will you use to digest the vector? The sequence of the restriction site and position of cleavage for each enzyme is shown below. So from the options we have to see which type of enzyme creates a fragment like this. That is G that is you will get a fragment like this. Okay, my drawing is very bad with computer, but still I am trying to make you understand. Okay, so if you see option C, you can see that there is an A and so this fragment, that is option C, it will be complementary to that or with that of the gene of interest. For ligation to occur, there should be complementary base sequence between the vector and the gene of interest. So here you have got GATCC. Now here also GATC is there. That is complementary to CTAG. 1 minute. See here, it is GATC. So that is complementary. When you take this portion over there, you can see that GATC will be complementary to CTAG and it can bind over there with complementary base pairing. So option C will be the answer. Answer is option C. Then next one. Which one of the following is not important for regulation of the tryptophan operon by attenuation? So presence of two adjacent codons for tryptophan in the leader peptide sequence. Coupled transcription and translation. Concentration of tRNA charged with tryptophan. The operator sequence of the tryptophan operon. So answer is which of the following is not important for regulation of tryptophan operon by attenuation is the operator sequence of tryptophan operon. Then next one. A plasmid DNA when digested with EcoR1 gave a single band of 16 kb. When the same plasmid was digested with BAMH1, it gave two bands of 6 kb and 4 kb. The plasmid has. So this is your plasmid. When you digest with EcoR1, so it has got only a single restriction site. It will give a single band like this. But when you digest it with BAMH1, you got two bands. One is 6 kb and the other is 4 kb. But what is the size of the plasmid? It is 16 kb. So 4 kb plus 6 kb, it will be only 10 kb. The rest 6 kb also will be digested and it will be released as a band because BAMH1 it will have 3 restriction sites. So answer will be option B, that is single site of EcoR1 and 3 sites of BAMH1. Only then you will get 6 kb, then 4 kb and an other 6 kb also. These two bands, they will be seen in the gel as 6 kb, the two bands, they will be seen as a single band. because it is 6 kb. Both the bands are 6 kb, so it will be seen as a single band, whereas the 4 kb band will be separate. So you will get only 2 bands, but there is a third 6 kb also. Only then the plasmid size will come up to total of 16 kb. Okay, so the answer is option B. Then myeloma cells fused with spleen cells in hybridoma technology are. So they are immortal antibody producing cells. A, as well as option C, hypoxanthine guanine phosphoribosyl transferase lacking cells. So option A and C both are correct. Then enzyme inactivation by suicide inhibitors should be. So suicide inhibitors are irreversible inhibitors and they will be directed to active site. So answer will be option B, active site directed. Then resolution in adsorption chromatography is achieved at both the binding and elution stage. So answer will be option B. Then protein kinase phosphorylates protein at hydroxyl group of amino acid side chains. Which one of the following groups of amino acids contain side chain hydroxyl groups? So serine, threonine and tyrosine. Answer is option B. In a segment of a transcribed gene, the non-template strand of DNA has the following sequence. What will be the corresponding 5' to 3' sequence in the RNA produced from this segment of the gene? So RNA sequence will be same as the non-template strand, except that in place of T in DNA, it will be U in RNA. So the answer will be option B, that is AGCU CAC UG. Answer is option B. Then, if the DNA content of a diploid cell in the G1 phase of the cell cycle is X, then the DNA content of the same cell at the metaphase of meiosis 1 would be. So, there is a doubling of DNA content during the S phase. So, the answer will be option A, 2X. The alpha helix in a protein is primarily due to. It is due to intramolecular hydrogen bonds. Answer is option A. Amino acid with asymmetric C beta atoms, that is threonine, only 2 are there. They are threonine and isoleucine. This question is repeated and I have answered with explanation in some other question paper. I don't remember the question paper, but it is some PhD entrance exam question paper. You can refer it in my channel. Then what is the minimum number of edges that meet at every branch node in a phylogenetic tree? So this is the how a phylogenetic tree will look like. So you have got, this is the branch node, this one. 1 minute. This is the branch node. These are the branches. This one is the branch. So how many, now the minimum number of edges that meet at the branch node? So 1 over here, from here to here, 1, this one 2, and this one 3. So the answer is option C, that is 3. Then according to the induced fit theory, an agonist is defined as. So agonist is a compound that induces a specific conformational change in the macromolecule. Answer is option C. Then a recombinant protein is found to be expressed very poorly in E. coli. It is hypothesized that the expression is blocked at a translational step. The first experimental technique to test this is. So that is translational step. So it is during the protein synthesis. So quantitative option B. The experimental technique, the first experimental technique to test this is. Answer is quantitative RT-PCR. Western blot is for whether protein is present, but the expression itself is blocked at the translational step. So protein is not present. Then next one, EMSA is for protein DNA interaction studies and PCR is normal PCR reaction. So the answer here is option B, that is quantitative RT-PCR. Then next one. Three genes A, B and C that are functionally related. There is a point mutation in gene A due to which gene B is not expressed, resulting in a non-functional gene product C. What is the possible relationship between these three genes? So there are three genes, that is A, B and C, and three are related. So there is a point mutation in gene A due to which B is not expressed, resulting in non-functional gene product C. So C is the product, B is the gene. So A should be naturally a transcription factor for B, and B is needed for uh, production of C. So answer is option B, that is A is a transcription factor for B and B is needed for C to be functional. So answer is option B. Then next one. Detectable serum antibody against a T-independent pathogen is a good indication that. So it is a good indication that a functional B cell system exists. Answer is option A. So it is T-independent pathogen. So the detectable serum antibody against a T-independent pathogen. It is a good indication that you have got detectable serum antibody level. So it is a functional B cell system exists. Then, in an experiment, 4 different N-terminal blocked purified proteins were treated with glutaraldehyde individually. One of the protein did not get modified. What may be the reason? So, glutaraldehyde is one of the most effective protein cross-linking reagent. The glutaraldehyde cross-link is known to be highly non-specific process that involve most of the lysine residues distributed on the surface of the protein. So what they have asked, in an experiment, 4 different N-terminal blocked purified proteins were treated with glutaraldehyde individually, and one of the proteins did not get modified by treatment with glutaraldehyde. Now what is the reason why the protein did not get modified? So I told you that the protein glutaraldehyde crosslink is non-specific and it involves most of the lysine residues. So the protein lacks lysine. Answer is option C. Which one of the following signatures, the stretch of amino acids in a protein, will target it to the nucleus? So entry of large proteins into the nucleus require nuclear localization signal, that is lysine, 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 arginine, lysine inside the protein, near but not at the N-terminal end of the protein. So answer is option B, arginine, lysine, 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 arginine, lysine. Answer is option B. Then, which one of the following rearrangement is not permitted during somatic recombination in the heavy chain and light chain immunoglobulin loci? So, segments of genomic DNA within the immunoglobulin genes are rearranged in the cells of the B lymphocyte and the process is known as somatic recombination. So, recombination will only occur between gene segments located on the same chromosome, and it generally follows the rule that only a gene segment flanked by recognition signal sequence or RSS with 12 base pair spacer can be joined to one flanked by 23 base pair sequence RSS. That is known as the 12 by 23 rule. So 12 can be joined to 23 base pair RSS. Thus, for heavy chain, DH gene segment can be joined to a JH gene segment and a VH gene segment to a DH gene segment. So, DH can be joined to JH, VH can be joined to DH, but VH gene segment cannot be joined to JH gene segment. Directly, as both VH and JH gene segments are flanked by 23 base pair spaces and the DH segment have 12 base pair spaces on both the sides. So the answer is VH JH. It is not VL JL but VH JH. That is based on the 12 by 23 rule. Then next one. Junctional diversity in CDR23 during gene rearrangement results in the addition of. So in both heavy chain and light chain, the diversity of CDR3 is significantly increased by the addition and deletion of nucleotides at two steps in the formation of the junction between gene segments. The added nucleotides are known as P nucleotides and N nucleotides. Answer is option B, P and N nucleotides. Then next one. The function of negative selection of thymocytes in the thymus is to eliminate. So autoreactive thymocytes is thymocytes recognizing and reacting autoantigens or self-antigen. So answer is option D, that is autoreactive thymocytes. Then next one, antigen recognition by T cells in the absence of co-stimulation results in. So it results in T cell energy. So antigen recognition by T cells is there, but there is absence of co-stimulation. So it results in T cell energy. This is a repeated question. It has been asked for PhD entrance examination by various universities as well as by DBT. Then, mutation of homeotic genes often result in which one of the following developmental defects in Drosophila? So, homeotic genes are responsible for determining the identity of particular segment or structures of the body. So, mutation in homeotic genes cause displaced body parts such as antennae growing at the posterior end of the fly instead of the head. So, mutation in homeotic genes often result in which one of the following developmental defects in Drosophila. So it is option B, that is transformation of one segment to another. Answer is option B. Then next one. Transition type gene mutation is caused when? So transition is caused when a purine is replaced by another purine or a pyrimidine is replaced with another pyrimidine. So answer is option D. So AT is a replaced by GC. A is a purine, G is a purine, T is a pyrimidine, C is a pyrimidine. So answer is option D, that is AT is replaced by GC. Then which one of the following is not enriched in eukaryotic promoters located in active chromatin? Okay, so the answer here is option C, that is methylated cytosine. Now DNase 1 hypersensitive sites are regions of chromatin that are sensitive to cleavage by the DNase 1 enzyme. In this region, chromatin has lost its condensed structure, exposing the DNA and making it accessible. Histone acetylated histones. Histone acetylation may affect chromatin structure directly by altering DNA histone interaction within and between nucleosome, thus resulting in a more open, higher order chromatin structure. So the answer is option C, that is methylated cytosine. Then next one. One of the reasons why non-substrate inducers, that is IPTG, are preferred over substrate inducers, example lactose, for induction of an inducible operon is because they directly interact with repressor and prevents repressor binding to the operator, thereby allowing RNA polymerase binding and transcription and translation of structural genes in the case of lac operon. So answer is option B. They directly interact with the repressor. Then in humans, enzyme having reverse transcriptase activity is, so it is telomerase. Answer is option D. Then next one. To prepare a DNA probe of high specific activity for detecting a single copy gene in a Zoo-blot DNA experiment, which one of the following procedures would be preferred? So you have to prepare a DNA probe for use in Zoo-blot experiment. So this can be done by using random primer labeling. So how is this done? So first you have to denature and anneal in the presence of a mixture of random hexanucleotides. So first DNA is denatured, then it is annealed in the presence of random hexanucleotides like this. Then clean-out subunit of the DNA polymerase plus DATP, DCTP. If you want to label with at the cytosine residue, then you have to use labeled DCTP, then DGTP, DTTP. So these are added after denaturation and annealing in the presence of random hexanucleotides. Clean-out subunit and the various DNTPs are added, and one of the DNTP is radio labeled. So what will happen? The random primers can then be extended using DNA polymerase, that is the clean-out subunit, and incorporating the label nucleotides. So that is how random primer labeling can be used to prepare DNA probes. Then the function performed in the smooth endoplasmic reticulum is biosynthesis of secretory proteins, folding of membrane proteins, addition of N-linked sugars, and detoxification of sugars. So four common functions of the smooth endoplasmic reticulum are mobilization of glucose from glycogen, calcium storage, drug detoxification, and synthesis of lipids. So out of these, drug detoxification is the answer. That is what is given in the options. Then next one. In human carcinomas, many proteins including most cytoskeletal proteins undergo modifications, thereby making it difficult for a clinician to identify the origin of the cancer cell. In this context, which one of the following can be relied upon for the identification of origin of cancer cells? So when normal cells are transformed to cancer cells, original signature expression of intermediate filaments are largely retained. So for the identification of the origin of cancer cells, intermediate filaments can be relied upon. Answer is option D. Then most organisms can regulate membrane fluidity by changing the lipid composition. If cells are transferred from a warm environment to a cold one, it can be expected that. So membrane containing lipids with longer fatty acid tails are less fluid than membranes containing lipids with shorter fatty acid tails. So membrane consisting of long chain lipids have higher melting temperatures than membrane consisting of short chain lipids. So in order to maintain fluidity of the membrane, there should be increase in the proportion of short chain fatty acids than long chain fatty acids. So the answer is option B. There will be an increase in the proportion of 16 carbon fatty acids compared to 18 carbon fatty acids. Then next one. Glycosylation of membrane proteins and lipids is carried out by enzymes present in the lumen of endoplasmic reticulum and Golgi. The glycosylated part of the membrane protein and lipids in the plasma membrane is likely to be. So it is likely to be exposed to the extracellular environment. Then next one. Lysosomal storage diseases are a group of inherited diseases that are characterized by accumulation of specific substances or class of substances within the lysosomes. All of the following mechanism can cause lysosomal storage disease except defects in the enzyme N-acetyl-glucosamine phosphotransferase. It causes eye cell disease or mucolipidosis too. Then defect or missing acid hydrolases, defects in the transport of lysosomal enzyme to the surface. That is the Answer, except they have asked all of the mechanism can cause lysosomal storage disease except defects in the transport of lysosomal enzymes to the cell surface. So coming to the next question, the gene encoding an enzyme A that functions in a metabolic pathway for conversion of metabolite X to Y was knocked out, but it still resulted in the formation of metabolite Y. From this it can be concluded that the gene enzyme encoding an enzyme A is required for the conversion of X to Y. Now that gene was knocked out. That means it was removed. I can say it is actually knocked out. That is the original terminology, knocked out. But it still resulted in the formation of metabolite Y. Now from this we can conclude that the enzyme A is sufficient but not necessary for the formation of Y. Then next one. 4 yeast mutants block membrane and secretory proteins in following compartments. So mutant A is blocking in the Golgi, B in the endoplasmic reticulum, C in the cytosol and D in the secretory vesicle. If 2 mutants are made that have combined effects A plus C and B plus D, the proteins of the new mutant will accumulate in. So protein cargo, it moves from the endoplasmic reticulum to the Golgi and is modified within the Golgi and is then sent to various destinations in the cell including lysosomes and the cell surface. Now A plus C is mutant A that is in the Golgi. So it will remain in the Golgi or else it can be secreted to the cytosol. But in the second case, that is B plus D, it is the mutant B that is endoplasmic reticulum. So it will remain in the endoplasmic reticulum itself. So here 2 answers are there, that is Golgi and endoplasmic reticulum or cytosol and endoplasmic reticulum respectively. Answer is option A and C. Then next one. The following shows the electrophoretic migration of a secretory protein in SDS page from the time of its synthesis, that is 0 minutes, to its secretion 90 minutes from the cell. Assuming that there is no problem in the sample preparation and SDS page, the reason for the lower band at 30 minutes and a higher band at 60 minutes and 90 minutes compared to that of the 0 minute is due to. So it can be due to any post-translational modification like phosphorylation of serine, threonine and tyrosine, or sulfation of tyrosine, or even glycosylation. It will slow down the mobility of your protein in the SDS page. So the answer here is the protein underwent processing and post-translational modification as a function of time. So in the beginning it was at time 0 minutes it was not modified, but later it got modified by phosphorylation or glycosylation or sulfation, and it resulted in the slowing down of the mobility of the protein in the page.